Okay, this is going to be a shorter one on hATTR amyloidosis and how it can be treated, building on some of the information we went over last time on RNA interference. Before we go on to a more dense topic of CRISPR systems and how they were discovered, how we use them for genetic engineering. So this is just going to finish off the last bit on RNA interference that we did last time before we move on to CRISPR. So hATTR amyloidosis is caused by a mutation in the gene coding for a protein called transthyretin, the TTR gene, which impacts the protein's function. Transthyretin, produced by the TTR gene, is mainly produced in the liver to help transport thyroid hormones and vitamin A around the body. The mutation in this TTR gene, which causes hATTR amyloidosis, results in the transthyretin protein becoming malformed and dysfunctional, becoming an amyloid protein. These amyloid proteins will accumulate in the body's tissues and cause damage, the same way as amyloid proteins accumulate in nerve cells in Alzheimer's. So these amyloid proteins, they accumulate over time, so the sufferers tend to be adults and the condition is degenerative, so it gets progressively worse over time. The amyloid proteins accumulate in the heart, peripheral nerves, and eyes and the kidneys in hATTR amyloidosis, causing tissue damage. Protein accumulation in these tissues can lead to organ failure and eventually death if untreated. Now, thanks to modern genetics, there are a few treatments for hATTR amyloidosis, which is a fairly rare and a hereditary disease. So it can be treated via the use of gene silencing therapy, reducing the abnormal amounts of the TTR gene transcripts that produce the gene products that accumulate to form the amyloid deposit. So there are multiple ways to silence this gene. One of them is CRISPR, where the gene is essentially cut out and replaced with a functional version of the gene. We're going to go more into CRISPR in the next video. Epigenetic silencing, where we can kind of prevent expression of the TTR gene by wrapping it around a nucleosome, so it's not in its transcribable form of what was the word now that we went over last time? Euchromatic genome. So the DNA is not in its kind of euchromatic form. It's bound around the nucleosome, so it can't be transcribed and it can't be translated, like the ribosome. Multiple ways for this epigenetic silencing. Easiest one to talk about: methylation, tagging that gene to the nucleosome so it can't be unwound by the DNA helicase and then transcribed by an RNA polymerase. Deacetylation and histone tail modification could also work, but I think they'd kind of be harder for a doctor or whoever is treating you to actually administer. I think the most likely would probably be methylation, but I'm not much sure. You can look it up yourself if you want to know. The main one relevant to the last video we talked about is RNA interference. a little about RNA interference in humans compared to in plants, because last time we were talking specifically about how RNA interference works in petunias. So it's basically the exact same, except for in humans, instead of having small interfering RNAs form complexes with the risk, we have microRNAs form complexes with a risk, but it's the exact same thing. MicroRNAs control around 30% of the genome, 10,000 genes, all the coding genes. There is only about 300 of them, I believe, complementary to specific sites. To treat it, we just introduce more of the microRNAs complementary to the TTR gene. These are going to form complexes with a risk. A risk is going to cleave the TTR gene transcripts, so they can't be translated, so we're not getting production of any of the mutated transthyretin protein, and no buildup of amyloid deposits causing the tissue damage, organ failure, and death. A bit about microRNAs. Like I just said, very important to regulating gene expression. There are 300 microRNAs in humans. Each microRNA can target several genes. Each gene can be regulated by several microRNAs. So if we were to introduce more of the mRNA that regulates the mutated TTR gene in hATTR patients, then we can activate the RNA interference that we want with the specific mRNA for the TTR gene, forming a complex with a risk. The risk complex is going to then be complementary and recognize any transcript of mRNA coming from the mutated TTR gene. It's going to pick up that mRNA and cleave it, so it can't be translated to produce the mutant transthyretin, and so no transthyretin can accumulate into the amyloid deposits that cause the degenerative effects of hATTR amyloidosis. Hopefully that made sense. It's basically just saying, last time we went over how RNA interference works in petunias. This gives a kind of case study kind of example about an example of microRNA interference in humans, because we can treat hATTR amyloidosis with this specific RNA interference. But yeah, humans are basically the exact same. We'll recognize a product that's, if in our cells there's an unusual amount of some kind of mRNA, we'll get our RNA-dependent RNA polymerase. It will form an antisense strand to form a double-stranded RNA. That's going to get cleaved by dicer to produce our microRNAs that are complementary to the mRNA that we want to get rid of and cleave because we think it might be pathogenic. So yeah, dicer cuts the double-stranded RNA into the microRNAs, or they may still be small interfering RNAs in humans. I'm not massively sure how they're different. Maybe microRNAs are kind of just more of a regulatory molecule that we have that will also kind of pick up and be complementary to the genes that they're targeting. That would probably make the most sense. But either way, humans also have small interfering RNAs and have the exact same process, just less often. Most of the time it's microRNAs in humans that are kind of being used in the RNA interference. So that's all.